Hello, my name is Caitlin Garmer, and today I'll be discussing my systematic review on the impact of Mediterranean diet on colorectal cancer recurrence following chemotherapy treatments. A brief introduction about me. I have my Bachelor's of Science degree in biology from Grand Canyon University, which I attained in 2016. I am currently a Master's of Science in Applied Nutrition candidate at the University of New England with an expected graduation date of August 2020. Some of my previous work experience, I have worked as a caregiver for the elderly, where some of my main duties have included meal planning, daily activities, and daily hygiene. And I've also worked as a medical scribe where I've followed a hand surgeon and written his patient notes. My volunteer experience includes volunteering for Grand Canyon University AZ HOSA. This was a Future Health Professionals Club. Some of the duties I had there was I worked as the vice president and I oversaw the community service branch where I was involved in community outreach and participating in community activities. And then one of my other volunteer experiences included being a research assistant at a lab called TGen, where I assisted with cancer research. Ultimately, my future career goal is to become a physician and utilize nutrition as the main uh, way of treating my patients. The objective of the systematic review is to assess the impact of Mediterranean diet on the rate of recurrence following colorectal cancer treatment. So I'll begin with some brief background information. So cancer tends to cause a lot of inflammation in the body, and colon cancer causes inflammation within the colon, resulting in tumor development. The innate immune system, adaptive immune cells, intestinal epithelial cells, and other stromal cells all play a big role in cancer-associated inflammation. So when inflammation begins, it disrupts the intestinal microflora, which is involved in homeostatic immune and metabolic function, and this is by metabolizing non-absorbed carbohydrates, dead epithelial cells, and mucus, while also producing metabolites that affect epithelial cell function, energy balance, and immune response. So when these are disrupted, this results in an increased risk of developing colorectal cancer. Short-chain fatty acids are suggested to promote the integrity of intestinal mucosa, and these are created through fermentation of non-digestible starches. The three most common short-chain fatty acids are acetate, propionate, and butyrate. Butyrate is a common energy source for colonic epithelial cells, and it has been shown to inhibit proliferation and suppress the growth of cancer cells. It's also been found to be one of the most important mediators for epithelial function and regulation. One study demonstrated that the group with the highest colonic content of butyrate had the lowest risk of colon cancer. In a follow-up study, low colonic short-chain fatty acids, along with high bile acids and high colon cancer risk populations were assessed, and fecal short-chain fatty acids were decreased in the population with a higher risk of colorectal cancer. So short-chain fatty acids are believed to play a very protective role in the gut microbiota, which has been shown to help reduce the risk of colorectal cancer. That leads us into discussing the gut microbiota. So the gut microbiota plays a role in maintaining the gastrointestinal barrier and maintaining intestinal homeostasis. Some of the more recent research has implied that the gut microbiota composition is associated with metabolic disorders and inflammatory bowel disorder. And the gut microbiota is greatly impacted by micro and macro intakes. Proper diet has a big impact on the gut microbiota. So when a diet is high in protein or fat with a decent amount of dietary fiber, short-chain fatty acids can be increased and levels of beneficial microbes are restored, which then reduces the levels of toxic metabolites. So dietary fiber is an undigestible plant fiber, and this helps bind to potential nutrients and can help with nutrient absorption and metabolism. And then short chain fatty acids are a major product of microbial fermentative activity. So dietary fiber has been shown to counteract the earliest stages of colonic carcinogenesis, while resistant starches have been shown to provide a protective effect to the DNA to prevent alterations leading to colorectal cancer. So now to discuss the components of the Mediterranean diet. So the Mediterranean diet is high in fruits and vegetables, fish, whole grains, olive oils, with limited amounts of dairy, alcohol, and wine. So the Mediterranean diet and colorectal cancer. So starting off with fruits and vegetables. Fruits and vegetables have a high content of flavonoids, which may have an antioxidant, anti-inflammatory, anti-mutagenic, and anti-proliferative properties. Fish, which is one of the main protein sources in the Mediterranean diet, is high in omega-3 fatty acids. Omega-3s are believed to suppress carcinogenesis and can be a protective factor against tumor initiation and progression. They've also been shown to integrate into the plasma membrane of cancer cells and inhibit signal transduction to promote apoptosis. Whole grains increase dietary fiber intake. Fiber reduces stool transit time, which potentially could reduce the amount of cancer-causing cells in the colonic epithelium. In a study assessing fiber intake and risk of colorectal cancer, there was an inverse association between risk of colorectal cancer and total fiber intake. And this is due to the belief that fiber dilutes fetal carcinogens, shortens fecal transit time, and reduces contact of carcinogens in the colon epithelium. Olive oil is high in polyphenols, which reduces oxidative damage to cellular DNA. The antioxidant activity of olive oil in colorectal cancer can help block tumor formation, inhibit proliferation and invasion, induce apoptosis, and regulate inflammatory response. And olive oil is the primary source of fat in the Mediterranean diet, so this would be consumed in higher quantities. Overall, the Mediterranean diet has shown a 13% decrease in cancer mortality. The belief is the anti-inflammatory properties of the Mediterranean diet can help reduce the risk of colorectal cancer. Many of the studies that have been reviewed have been conducted prior to colorectal cancer development, but the belief is this would translate to after treatment for colorectal cancer. Moving on to the methods of the systematic review. 3 databases were searched for the systematic review: PubMed, the UNE library, and Embase from January of 2010 to June of 2020. The search terms included chemotherapy, colorectal cancer treatment, Mediterranean diet, Medi diet, adults, and clinical trials. And the limitations included research trials, cohort studies, randomized control trials, and case studies. So the inclusion-exclusion criteria, to be included, it had to be a research-based article. It had to be in English from 2010 to 2020. The research had to include adults over the age of 18. They've had to receive treatment for colorectal cancer, and it had to be a clinical trial. Exclusions included published before 2010, included participants under the age of 18, were not available in English, animal studies, or they were meta-analysis or systematic reviews. So through database searches, 72 articles were identified and screened. 22 articles were excluded based on publication date. The remaining 50 articles were assessed for eligibility, and 35 were excluded due to animal-based trials, prospective studies, systematic reviews, and they addressed additional cancers. The remaining 15 articles were included in the systematic review, and the primary data extracted were outcomes, confidence intervals, and p-values to compare statistical results. All were assessed for quality and bias using the AND-EAL rating. So the results of the systematic review. The results have been broken up into 6 different sections. So the first section is lifestyle factors, and there was one research trial that assessed lifestyle factors. It was a case control study with 1,014 participants. And the results of this study found that patients who are diabetic, are smokers, are obese, have a familial history of colorectal cancer, lower physical activity rates, and consume greater amounts of alcohol have a greater risk of developing colorectal cancer than controls. In addition, dietary patterns that closely aligned with the Mediterranean diet guidance were associated with mediating the adverse effects of the unhealthy conditions when adherence was medium to high. As we can see on this table, down at the bottom on the Medi-Diet score, the medium and high adherence both had p-values under 0.05, which gave them statistical significance. The next group of research trials can discuss the components of the Mediterranean diet. So starting with a case control study that was based in Saudi Arabia, consumption of dairy products, legumes, leafy vegetables, olive oils, black tea, and coffee were assessed weekly in 137 colorectal cancer patients. Protective diet associations were seen with 3 to 5 servings of legumes, leafy vegetables, and olive oils, which are all part of the Mediterranean diet pattern. So fish is one of the main staples of the Mediterranean diet. So marine omega-3 polyunsaturated fatty acids were assessed in a study that assessed the diet of 1,011 colon cancer patients during and following chemotherapy treatments. Marine omega-3 polyunsaturated fatty acids consumption were assessed to determine its relationship with disease-free survival. And the results showed that a greater than or equal consumption of 0.15 grams per day were associated with a 70% lower risk of mortality following colorectal cancer treatments. On the contrary, the previous mentioned case study in Saudi Arabia showed no significant difference between colorectal cancer patients and the control groups with regards to seafood consumption. Lastly, a trial assessing nutritional behaviors in 500 patients revealed that the Mediterranean diet adherence is associated with a 13% lower odds of having colorectal cancer, and coffee consumption, added table salt, and non-stick cookware were associated with colorectal cancer.